2A student investigated the digestion of starch. Fig. 2.1 shows the apparatus she used. Dialysis tubing is used to represent a cell membrane. The dialysis tubing material allows small molecules to move across it, but not larger molecules. Test tubes A and B were set up as shown in Fig. 2.1 and placed in a water bath at 37 degrees Celsius for 30 minutes. The liquid outside the dialysis tubing in test tubes A and B was tested with Benedict's solution at 0 minutes and after 30 minutes. Table 2.1 shows the results. A. Using the information in Fig. 2.1 and Table 2.1, explain the reasons for the difference in the results for test tubes A and B in Table 2.1. Benedict's solution in A remains blue, no color change, negative result, as there is no maltose breakdown of starch. Benedict's solution in B turns red, changes color, positive result, in the presence of maltose. Amylase breaks down starch into maltose. Maltose is a small, soluble molecule. Maltose is able to pass through the dialysis tubing by diffusion from an area of high concentration to low concentration down a concentration gradient. B. Complete Table 2.2 by writing in the names of the missing enzymes, substrates and products. C. State the name of the structures that increase the surface area for absorption in the small intestine. One. Villi, microvilli. 0610_W22_Ms_4 extended 3. 2. 8. Digestive enzymes are secreted into the alimentary canal. I, as well as enzymes, other substances that are important for digestion are also secreted into the alimentary canal. State the names of 2 of these other substances. Any 2 from bile, hydrochloric acid, mucus, water, saliva. E. Table 2.1 summarizes some facts about the enzymes that are secreted into the alimentary canal. Complete Table 2.1. B. The activity of the enzyme pepsin was measured at different temperatures. The results are shown in Fig. 2.1. Describe and explain the effect of temperature on the activity of pepsin. Use the information in Fig. 2.1 to support your answer. Any 6 from 39 plus or minus 2 is optimum temperature. Highest activity is 2500 U per milligram protein. Steeper decrease in activity after optimum. No activity after 62 degrees Celsius plus or minus 2. Increased temperature causes increased kinetic energy. Increased frequency of effective collisions. Forming enzyme substrate complexes. Denaturation occurs at any temperatures above optimum temperature. Active site changes shape. No longer fit into substrate, complementary to substrate. Idea that both enzyme and substrate are proteins and may change. 6. Each unit must be stated at least once. Don't accept on wrong or no units. C. Lactase is another digestive enzyme, which is produced by human babies. I. Explain why lactase is important for the nutrition of young babies. Any 2 from. Lactose is found in milk. Lactase breaks down lactose. Young babies are dependent on lactose milk for their nutrition. E. Most people stop producing lactase when they become adults. If they drink milk, this may give them diarrhea. Describe the symptoms of diarrhea. Loss of watery feces. E. Cholera can also cause diarrhea. Explain how the cholera bacterium causes diarrhea. Any 3 from. Cholera bacterium release toxins. Causes chloride ions to be secreted from the body. Chloride ions in small intestine, duodenum, ileum. Ref to osmotic movement lowers water potential in gut. Water moves into gut or more water remains in intestine's gut. 0610_S23_Ms_41. 1b. A student investigated the effect of bile on the digestion of fat in milk. They set up 3 different test tubes. Test tube A contained milk and bile. Test tube B contained milk and lipase. Test tube C contained milk, lipase and bile. They used an indicator that is pink in alkaline solutions and colorless in acidic solutions. They added the same volume of indicator to each test tube. The student observed and recorded the color of the contents of each test tube at 0 minutes, 20 minutes and 40 minutes. Table 1.1 shows the results of the investigation. I explain the results for test tubes B and C in Table 1.1. Any 4 from pH decreases, solution becomes acidic. pH changes because fatty acids are produced. Lipase digests, breaks down fat. Fatty acids produced faster in test tube C than B. Bile emulsifies fats, converts large particles of fat to small particles. Bile increases the surface area for lipase action. Explain the purpose of test tube A in Table 1.1. Compare with tubes B and C to assess effect of lipase and/or bile. Shows that bile does not chemically digest fats, does not make solution acidic. Shows that lipase enzyme is required for breakdown of fats in milk. Control experiment. See, the action of lipase is affected by temperature. Fig. 1.2 shows the axes for a graph of the effect of temperature on the activity of lipase. Complete the graph by drawing a line to show the expected effect of temperature on the activity of lipase. Adding a label line and a label to show the point at which all the lipase has been denatured. Line drawn showing that decrease after optimum is steeper than increase. MP1 line does not have to start at the origin or end at the x-axis. Label line to point where line meets the x-axis and label indicating that all the lipase is denatured. MP2 line must meet the x-axis. Explain why lipase cannot be used to catalyze the breakdown of proteins. Any 3 from ref to specificity. Only substrate for lipase is fat molecules. Shape of active site is not complementary to protein, complementary to fat. Protein cannot fit into, bind to active site lipase enzyme. Enzyme substrate complexes cannot be formed. 0610_S23_Ms_42. 7. Complete the sentences about enzymes by writing a suitable word or phrase in each of the spaces provided. Enzymes are involved in chemical digestion, which produces small, soluble molecules that can be absorbed into the blood. 2 examples of protease enzymes are pepsin and trypsin. Pepsin is produced by the stomach and requires acidic conditions. These conditions are created by the release of hydrochloric acid, which provides the optimum pH for pepsin activity and also kills harmful, named microorganisms, pathogens. The pancreas, small intestine, produces trypsin, which breaks down protein in alkaline pH conditions. These conditions are created by a substance called bile salts, which neutralizes the gastric juices and also has an important role in the emulsification of fats and oils. 8. 0610_S22_Ms_42. 6. 8. Complete Table 6.1 to show the names, functions and sites of action of the 3 different digestive enzymes. B. Enzymes are proteins. Fig. 6.1 shows the stages involved in protein synthesis. I. State the name of the parts represented by the letters A and C in Fig. 6.1. A gene, allele. C. Ribosome. E. Describe the events that occur during stage 2 in Fig. 6.1. mRNA, a copy of the gene array, moves out of the nucleus to the cytoplasm. E. State what determines the order in which the parts labeled B are assembled. The sequence of bases in mRNA, DNA, gene. C. The shape of a protein is very important for its function. Explain the importance of shape for the function of an enzyme. Ref. To specificity. Shape of active site and substrate must be complementary. To enable enzyme and substrate to bind, enzyme substrate complexes to be formed. 0610_S22_Ms_41. 1a. Some students were studying the activity of yeast. They made a fact file, as shown in Fig. 1.1. I. State the kingdom in which yeast is classified. Fungus, fungi. E. State the process that occurs in mitochondria to provide energy for yeast cells. Aerobic respiration. B. Yeast cells make the enzyme sucrase. Sucrase catalyzes the breakdown of sucrose to glucose and fructose. Enzymes are made of protein. Explain how the shape of a sucrase molecule is related to its function. Any 2 from ref. To specificity of enzyme. Shape of active site is complementary to substrate, sucrose. 4. Substrate, sucrose, to bind, fit, enzyme, sucrase, active site. C. The students made an extract of sucrase from yeast cells. They investigated the activity of the sucrase extract at different pH values. They determined the rate of reaction at each pH. They then calculated the rate of each reaction as a percentage of the fastest reaction, to give the percentage activity of sucrase. The results of this investigation are shown in Fig. 1.2. Describe and explain the effect of pH on the activity of sucrase shown in Fig. 1.2. Total of 6 from max. 4 for description. Sucrase enzyme is active between pH 2 and pH 12. Activity increases and decreases, reaches a peak. Peak maximum or 100% activity at pH 6. Steeper increase between pH 5 to 6, steeper decrease in activity between pH 9 to 10. Minimum 10% activity at pH 12. More activity in acidic conditions, low pH, than alkaline conditions, high pH. Explanation. Change in pH affects the shape of sucrase active site enzyme. At pH 6, most enzyme substrate complexes form. At low-high extremes of pH, enzyme is partially denatured. Ref to substrate molecules can no longer bind with enzyme at low-high extreme pH, so activity decreases. 0610-S21-Ms_43. 1a. The main component of milk is a liquid that is an important solvent in the body. State the name of this solvent. Water. Table 1.1 shows some of the nutrients found in human milk, the elements that make up the nutrients, the enzymes that digest them, and the products of digestion. Complete Table 1.1. Beet milk also contains vitamins and mineral salts. I explain why vitamin D is important for the nutrition of children. Any 3 from needed for growth development, strengthening of bones. Needed for growth development, strengthening of teeth. Prevents rickets. Explain why iron is important in the diet. Any 2 from required to make hemoglobin, red blood cells. For transport of oxygen. Prevents iron deficiency anemia. C. Rubella and kwashiorkor are 2 diseases that affect children. Rubella is a transmissible disease and kwashiorkor is a non-transmissible disease. I define the term transmissible disease. Disease is caused by a pathogen. Pathogen is passed from one host to another. Explain why women should be vaccinated against rubella before they reach reproductive age. 1 CIINE 4 from rubella is caused by a virus. Virus can cross the placenta. Virus can affect embryo fetus. Can cause serious disease, damage, death to baby offspring. Vaccination stimulates long-lasting active immunity in the mother. Immunity gained before they become pregnant. Outline the causes of kwashiorkor. Not enough protein in the diet. Adequate energy in the diet. Inadequate diet after breastfeeding finishes. 0610-S20-Ms_41. 2 biological washing powders contain enzymes that break down food stains. A complete Table 2.1 by naming the enzymes that break down 3 substances in food stains and by stating the product or products. Some students compared how effective biological and non-biological washing powders are at removing stains at temperatures between 10 degrees Celsius and 60 degrees Celsius. Pieces of stained cloth were washed using 2 different washing powders. The degree of stain removal was measured by using a light meter to record the percentage of light reflected from the cloth. A light meter gave a value of 100% when the cloth was completely clean. Any stain left on the cloth reduced the percentage of light reflected. The results of the students' investigation are shown in Fig. 2.1. B. Compare the effectiveness of the 2 washing powders at removing stains. Use the information in Fig. 2.1 in your answer. Any 4 from biological washing powder is more effective at lower temperatures between 10 degrees Celsius and 40 degrees Celsius, 10 degrees Celsius and 43 degrees Celsius. Comparative data quote for the difference at a stated temperature. Biological washing powder removes all stain between 30 degrees Celsius and 40 degrees Celsius. Non-biological removes all stain only at 60 degrees Celsius. Effectiveness is similar at high temperatures between 50 and 60 degrees Celsius. Same trend, below 30 degrees Celsius, at low temperatures, from 50 degrees Celsius. Idea of effectiveness of biological washing powder decreases between 40 degrees Celsius and 44 degrees Celsius, no such decrease for non-biological washing powder. C. The students suggested that the enzymes in the biological washing powder were denatured at high temperatures. Explain why enzyme molecules do not function when they are denatured. Any 2 from active site changes shape. Substrate no longer fits into enzyme active site. No enzyme substrate complex, no successful collisions. D. Forensic scientists often try to find DNA on items of stained clothing. The DNA can be used to identify individual people. suggest why DNA can be used to identify individual people. Any two from individual people have different, unique DNA. DNA has genes or alleles have sequences of bases.